In my video, I will be discussing the protein Prestin. Prestin is a protein located in outer ear hair cells and functions to help amplify sound waves for detection by inner ear hair cells. What makes Prestin unique is that it has the ability to change size depending on the charge and voltage of its environment. When many of these Prestin molecules expand and contract in the outer ear hair cell membranes, the entire hair cell actually changes in size as well. When these size changes are rhythmically in sync with incoming sound waves, amplification will occur. Here's a short clip illustrating this process.
1, 2, 3 o'clock, 4 o'clock, rock. 5, 6, 7 o'clock, 8 o'clock, rock. 9, 10, 11 o'clock, 12 o'clock, rock. We're gonna rock around the clock tonight.
While that looked rather silly, it actually was a hair cell responding to the song and moving due to the expansion and contraction of Prestin. Before we delve into the biophysics of this process, I want to start with a little background on auditory processing in the ear. As I'm sure you all know, sound travels in waves of compressed and expanded air in space. As these waves enter the ear, they cause what is called the tympanic membrane to vibrate in sync with the sound wave. These vibrations cause special ion channels in inner ear hair cells to open and close in sync with the vibration of the membrane and the auditory input wave. The opening and closing of these channels cause different electrical currents into and out of the cell, in turn creating a voltage change cycle in sync with the sound wave's frequency. These voltage changes are what lead to neurons firing and auditory processing in the brain. Incoming sound waves from the outside world would not be loud enough to result in neural activity and recognition of a sound. Thus, mammals have developed several mechanisms for amplifying sounds within the ear to an audible level. Among these is this Prestin process. Just as sound waves cause the tympanic membrane and inner ear hair cells to vibrate in phase, they also cause what is called the outer ear hair cells to vibrate in sync. Like the inner ear hair cells, this vibration causes special ion channels to open and close rhythmically in the outer hair cells. The result of this is charge rushing in and out of the cell, and thus voltage changes occurring in the same rhythm as the incoming sound wave. Because Prestin responds to local voltage changes, Prestin's rhythmic expansions and contractions are also in phase with the sound wave. This motility within the outer ear is what causes the mechanical amplification of the sound wave. For example, at high pressures from the sound wave, outer hair cells depolarize, resulting in a Prestin contraction. At low pressures, outer hair cells hyperpolarize, resulting in a Prestin expansion. Continual expansion and contraction actually inputs mechanical energy into the sound wave, resulting in a larger amplitude and thus a louder sound. In order for Prestin to change size, it clearly must form different conformations, each taking up a different amount of space. The exact mechanism is not yet known, but there are several proposed models. Studies agree that there are at least 2 voltage-dependent steps involved in the size change. It is believed that there is an intrinsic and extrinsic voltage sensor for Prestin. The location of the intrinsic voltage sensor is not yet known, but there are several proposed regions, such as the alpha helix dipole that places partial positive charge near the N terminus and partial negative charge near the C terminus. Also, it is possible that various charged amino acid residues are contributing to the voltage sensor. When all amino acids were made neutral, voltage sensitivity was partially attenuated, but not fully, most likely due to the continued presence of the alpha helix dipole. Intracellular anions, particularly chloride, have been shown to act as an extrinsic voltage sensor and allosteric modulator to Prestin. These ions have been shown to be necessary for voltage sensitivity. Changes in voltage across the membrane create an electric field, causing chloride anions to move. It has been shown that chloride will actually move from the cytosol into the protein, toward the extracellular end, when the cell is hyperpolarized. Chloride moves back out as the cell is depolarized. Chloride's presence within Prestin, coupled with charged regions of the peptide reacting to the electric field, will induce a change in conformation of Prestin and thus a change in the size of the hair cell. Prestin belongs to the SLC26 family of proteins. These proteins are, for the most part, an anion transporter family. This helps to explain chloride's role in voltage sensing modulation. Chloride is able to enter the protein because it is of a family of proteins which allow anions to pass through them. Unlike other proteins in the family, however, Prestin has an incomplete ion channel, such that chloride may enter the protein from the cytosol, but not pass through it. Orthologs of this protein in non-mammals all serve for divalent anion transport. However, electromotility is only found in mammals. Mammalian and chicken isoforms have very similar sequences, especially in the hydrophobic region of the protein which makes up the channel. This helps show that mammalian Prestin most likely evolved from an anion transporter to develop this electromotility that we now see. Studying Prestin is very important, as it seems to be a major cause of deafness in mammals. In one experiment,
Knockout mice were created without the gene coding for Prestin. These mice were rendered deaf, confirming Prestin's role. If we could find a way to reinsert Prestin into human outer hair cells, perhaps we could find a way to reverse deafness in many patients. Prestin has also been shown to be crucial for ultrasonic hearing organisms such as bats and whales. Echolocation, for instance, seems to be based on a specifically mutated form of the Prestin gene. Ultrasonic hearing is a fascinating process, and further study of Prestin could teach us how animals in nature manage to code sound waves neurally so exactly.
There is still a lot more we have to learn about Prestin and the SLC26A protein family generally. We do not really understand the mechanisms of size transformation or even where the charge sensor is precisely located. We do not know the mechanism of anion transport in the family generally, or how chloride interacts with the Prestin protein. Research has come far in proving that Prestin does actually change size and changes the size of the hair cells with it. But before we can say we truly understand this process, there is much more work to be done.
1, 2, 3 o'clock, 4 o'clock, rock. 5, 6, 7 o'clock, 8 o'clock, rock. 9, 10, 11 o'clock, 12 o'clock, rock. We're gonna rock around the clock tonight. Put your glad rags on, join me home. We'll have some fun when the clock strikes 1. We're gonna rock around the clock tonight. We're gonna